for inviting me to the Plant Health in Horticulture. I'm Dr Perez-Sierra. I am the head of the Tree Health Diagnostic and Advisory Service at Forest Research. Today I'm going to talk to you about pests and diseases. Then what is THDAS? We are the Tree Health Diagnostic and Advisory Service. This service has been running since the 1960s and is a team of pathologists and entomologists, and we provide impartial advice or identify pests and pathogens on trees in Britain. We have two main stations, one based in the south at Alice Holt and one in the north, just outside Edinburgh, and we work alongside the tree health teams from the three countries, England, Scotland and Wales. We also have the support of the devolved governments. Then how the advisory service, how THDAS fit in the bigger picture of tree health. In the last few years, we have an exponential curve on the number of pests and diseases they discover in the country. And this graph just shows you, in the circle, you can see the number of new pathogens discovered in the country and the insects. Then some examples that you will be very familiar with are horse chestnut leaf miner, or oak processionary moth, oriental chestnut gold wasp, elm zigzag sawfly, or the eight-tooth spruce bark beetle. If you have some example of pathogens, you probably are very familiar with Phytophthora ramorum or Phytophthora ostrothedri, Callara ash dieback, sweet chestnut blight, and we don't know what is coming next. Then we, from the advisory service, we try to gather information about the health of trees in Britain. We try to identify trends about pests and pathogens, how they occur or where they occur and how they spread. To have an indication of the most common disorders, to try to identify new threats to the British trees, we contribute to the management of pests and diseases in Britain. We work very closely with other agencies and other laboratories and with DEFRA. We provide advice to owners, managers, or relevant authorities. And how we see the advisory service is like an early warning system for tree health. Then, in a way, we work with common pests and diseases. We work on new and emerging pests and diseases. We work on the unknowns. This could be the new problems of tomorrow. We also do annual surveys on tree collections because they can give us an indication of what the problems might be. And we are involved also in different projects just to try to raise awareness on pests and diseases. And two of these projects are Observatory and Bridget. Then how does the advisory service work? We develop a system, it's a web-based system called Tree Alert. This system is available to anybody in Britain to report pests and diseases on trees. If you see anything of concern on any tree, you can report it through Tree Alert. Then, very important, before you use Tree Alert, you need to know what information you are going to need, because the system will take it from the first screen through a series of screens asking you for information, and it will be very disappointing to reach the end and you realize you don't have all the information required and you cannot submit your report. And before you submit any reports, I would recommend that you get the information that you need. If you are going to be a frequent user as well, I would recommend that you create your own account and you have your login in details, then you don't have to input your details over and over. Then within Tree Alert, you have different options. You can either submit a general report, and that means anything that you see on trees that is of concern, then you can report it. You can also submit a report on a specific pest and disease, and that is, we have different reporting lines. We have 5 at the moment. We have one for Callara ash dieback, one for Dothystroma needle blight. We have acute of decline, Phytophthora lateralis, and Asian longhorn beetle or citrus longhorn beetle. And then, what information is it required? We want information about you. We want your name, a valid email address because we may need to contact you. Also, we need information about your observation, the date when you observed the damage, the site, what type of site it was, the tree species, and the location. And normally we'll ask you for a grid reference. Also, we ask you about information about the problem you have observed. Is it in the crown, on the stem, on the base? And also photographs, 3 photographs. And this is very important. I really would like 3 photographs. One of the 3 in context, one of the symptom in context, and one of the details of the symptom. Then what happened to these reports? These 3 alert reports are checked daily, and they are allocated either to entomologists or pathologists. We give priority to regulated to quarantine organisms, because if we suspect one of these reports is a suspected quarantine organism, these reports can be followed up by the plant health services and authorities in England, Scotland, and Wales. In some cases, members of the THDAS will follow up these inquiries. We try to reply to all the inquiries we receive. It may take long, but we try to do it. And in some cases, a site visit will need to be arranged. And in some cases, we might request samples. Then what happened to the samples? If we request samples from you, you will send samples to us, to the laboratory, and we use a whole range of different techniques. From basic techniques, like we use microscopic examination, we could incubate your sample just to make sure that produce the fruit bodies or that we are looking for. We may do isolation on media. We do some baiting or trying to recover from soil some pathogens. We do DNA extraction, either from pure cultures or from the samples you are sending in. We sequence the DNA. We compare the sequences with databases, and we do, as I said, we do identification sometimes either by PCR or real-time PCR with either specific primers or not. And this is like the CSI, but on plants. And this is some pictures of my colleagues in the advisory team. Then this is who we are, what we do, and now we are going to have a look to the most reported pests and diseases through the advisory surveys, the advisory service last year. And the top 10 pest agents reported, you probably are not surprised that the top number 1 was oak processionary moth. And along this, you can see on this list a lot of caterpillars and moths, because often they are mistaken with oak processionary moth. Then in our first number 1 was oak processionary moth, followed by oriental chestnut gold wasp. Then we have different moths, as you can see here. Horse chestnut leaf miner is still on our top 10. And then we have more moths, as you can see, and green spruce aphid and elm zigzag sawfly. These were our top 10 pests reported last year. If we look to the diseases, and I am a pathologist and I will talk more about fungi, then we have our top number 1 was Xylella ash dieback. Even that it was detected in 2012, and we had every year has been number 1, and we think maybe will be a decline, still our number 1. Our number 2 is honey fungus. And what I'm going to do with all of this, I'm going to show you some pictures and talk you through. Then, as I said, number 1 is Xylella ash dieback, and we had a lot of cases last year. You can see in this map, the yellow dots are the samples detected in 2018, and the black dots are the ones detected in 2019. But also we have detected Hymenoscyphus fraxineus Xylella on other ash species. These are some of the ones we have detected, Xylella, and the ones in red is the ones where the fungus is also causing dieback of the twigs, where the ones in black we have only seen symptoms on the leaves and not in the wood. Also, we detected Hymenoscyphus fraxineus on different hosts, and these that they were not ash, they were not fraxinus species, but they belong to the Oleaceae family, and these are Filarea latifolia, the one at the top, and you can see similar symptoms as on ash. You have dieback of the twigs and branches, and also the discoloration of the wood, and also Filarea angustifolia and Chionanthus virginicus. As I said, our number 2 is honey fungus. Honey fungus will always be on the top 10 because it's one of the most common problems on trees. And you can see here some of the pictures of the fruit bodies, typical honey color mushrooms with the ring and in the collar, white spores. And when you look under the bar, you will find the white mycelium of the fungus. Our number 3 is Pseudococcus suli, and this is only a problem. It has been a problem since the end of 2013 and 2014 is when we started seeing this issue on cedars, especially on Atlas cedar. Although this fungus can also affect hemlocks, as you can see here, some pictures on Tsuga heterophylla and Tsuga mersensiana, but it's more dramatic on Atlas cedar, where it should be blue, and instead of blue, it turns pink in color. Phytophthora is always on our top 10 as well. We have a wide range of Phytophthora species detected. We have some in the soil, like Phytophthora cinnamomi. We have here, these are an example on pine or sweet chestnut. We have Phytophthora ostrothedri on juniper, lateralis on lawson cypress. We have a lot of Phytophthora bleeding samples coming through as well. You can see Phytophthora pseudosyringae or on beech or Cambivora, Plurivora on Tilia, Ramorum on oak, Illysis on holly. And these are, as I said, we have a whole range of Phytophthora causing different symptoms on trees in Britain. Diplodia shoot blight is another one on the top 10, and this was not in the top 10 a few years ago, but it's reaching this point of the top 10, and we are seeing more problems caused by Diplodia. Diplodia is a fungus, and it will cause, especially on pine, a shoot dieback, but also we have been finding Diplodia on other conifer species. Another emerging disease is Neonectia canker on fir, and this is a fungus that is not any fungus. This fungus has been around for a very long time, but in the last few years, it seems to be causing a problem on fir species. And the symptoms, you will see the early symptoms like dying of the branches. And if you look closer to the branches, you may see the cankers and even the red fruit bodies of the fungus, that when you have a close-up look to them, they look like red. They look like eggs of insects, but actually they are the fruit bodies of the fungus. Then when a tree is affected by Neonectia, you may see, as I said, dying of the branches, lots of resinosis on the trunk. This figure, the trees, especially on tree collections, some of these trees are removed because they look so unsightly. But also, you can see the picture there in the middle, it can affect also plantations. Heterobasidium root rot, this is probably one of the oldest and most well-known pathogens on conifers, and used to be known as fomes or fomes rot. And this is Heterobasidium annosum, but we also have detected another species, Heterobasidium abitinum, and at the moment we are doing some research on this species. Swiss needle cast is also another pathogen that we are detecting more and more. This is specific to Douglas fir, is caused by the fungus Nothophyocryptopus gaumannii, and basically you will see that the needles, the inner needles, they turn yellow and they fall as soon as you touch the branches. Mortality is rare, but it can cause growth reduction of up to 50%, and in severe cases up to 80%. The last one, the top 10, is powdery mildew. I think we're all familiar with powdery mildew. And this is a picture on plane, but probably you are very familiar on oak. It's very, very common on oak. Then this is a quick going through the top 10 pests and diseases reported through the advisory service. And now to finish the presentation, I'm just going to talk about Xylella fastidiosa. And this is not a fungus, this is bacteria. And it's not currently in the UK. And I want to make this very clear. So far as we are aware, it's not known to be happening in the UK, although it is in Europe. Then Xylella fastidiosa has been described as one of the most dangerous plant bacteria worldwide. It is insect transmitted, and can infect over 500 species, including crops, ornamentals, trees, and wild plants. We know in Italy alone, over 1 million olive trees are dying from Xylella. And as I said, so far, Xylella has not been reported in the UK. Then, Xylella fastidiosa is a xylem inhabiting fastidious bacteria. Fastidious is because it is very difficult to grow on media. This bacteria multiplies on the vessels of plants, the vessels become blocked, and water cannot reach the parts of the plant from the roots, and basically plants begin to suffer from like drought symptoms, and they wilt. And as I said, it's transmitted by xylem-feeding insects, and the meadow spittlebug, Philaenus spumarius, is the main vector identified in Europe as the primary, and is the primary vector in Italy. And this species is widespread in Europe, but it's also in the UK and feeds on hundreds of hosts. Then, common potential Xylella vectors in Britain. We have Neophylaenus lineatus, Philaenus spumarius, and Afrophora alni. They potentially could be Xylella vectors. Then, diseases caused by this bacterium, it range from crops to trees. For example, and you will find when you do any search on this pathogen that each disease has a different name. For example, on grapes is called Pests disease, but on peach is called phony peach disease. On plum is plum leaf scald. Then everywhere you look, you will find that different names of this pathogen causing disease on different hosts. But I want to show you what it can do to big trees. And this is an olive tree in Italy. And this is in March 2015. These red marks on the picture, it just showed the early symptoms. This was in September that year, and this was May 2016. This is just to show what this bacterium can do to a tree. Then there are different subspecies of Xylella fastidiosa, and the one that we probably were more worried about is multiplex. This is endemic to North America. They have numerous hosts and is associated to trees. And it's been found in ornamentals, on trees, and is one of the. We know this one happens in colder areas as well. Then, if we look into trees, the first report of Xylella on trees was on American elm, and then since the 1980s, it's been reported on different species, but especially on oak species, on plane, and on Acer. The disease is known as bacterial leaf scorch, is recognized as a major disease of street and landscape trees in the southeastern United States since 2010. The problem with Xylella is the symptoms on trees are not always distinct. In some cases, it can take a long time for plants to show any symptoms after being infected, or they might not show any symptoms at all. Symptoms are best observed towards the last part of the summer, and sometimes the problem is when you go towards the end part of the summer that the symptoms can be mistaken or confused with early senescence, because the symptoms that we are going to see, especially on trees, are leaf scorching, and you will see normally leaf scorch, marginal leaf scorch with a yellow halo, and then you may see defoliation or death of the trees. Then, if you have a look, for example, to the picture of the elm or the oak, or the bottom picture here, then you will see this can be confused very easily with early senescence. Then we, as part of the Bridget project, we have prepared some resources that can be very useful. We have prepared some leaflets where you can find information on the high priority or risk host for the UK and the symptoms. We have resources on identifying xylem-feeding insects. We have videos of where is Xylella, what is Xylella. You can find all the information in there. I will recommend These web pages is information on emerging risk, and is the UK Plant Health Risk Register, the Plant Health Portal. You can check the RHS for pests and diseases, and Forest Research for pests and diseases. Before I finish, just would like to say, remember, if you see something that you are concerned, please report it. We better identify what it is that means something that could be potentially important. And thank you very much for your attention.
Well, thank you very much, Anna. That was a really, really interesting presentation. There are a very few questions at the moment on the questions and answers box. So if anyone has a question and would like to pop it into the Ask a Question window, please do that now. And I'll start asking some of the questions that are there already. We have one from Kate Burtonwood talking about the Bufftip moth and saying it was one of the most reported pests or one of the most reported insects in the tree alert. So other than defoliation, she's asking, does this species present any other threat?
No, not really. I guess obviously what happened is because of OPM, we have so many reports in any other caterpillars, moths. We have so many reports, but yes, we are not very concerned with this one. But I'm not an entomologist. Tom, you are an entomologist.
I'm an entomologist, so I can say that it's not a major problem at all. It's a minor defoliator. And really the concern is that it just gets confused with the oak processionary moth caterpillar, which is something that we are very interested in tracking. So it's just an easily confused species. Okay, so I'll ask a question from me because I'm interested. Xylella is something that worries me a lot because I think it has a potential to cause a huge amount of damage to a wide range of plants. And you talked about marginal leaf scorch with the being a symptom. Is this a symptom of disease we need to avoid due to Xylella?
Sorry, Tom, but you are breaking off.
Yep.
I can't hear you well.
Okay, is this a symptom of any other disease? So we've got the leaf scorch with the yellow halo. Is it a symptom of anything else?
Okay. More than other disease, I would say is autumn color. If you go out now and you look to the change of colors in trees, in most of the trees, you will see the way the color breaks into the leaves. You have this brown marginal necrosis and also the yellow halo. Then because of the symptoms of Xylella are best observed towards the end of the summer, and obviously we have the beginning of the autumn, is many times the symptoms are confused with early autumn symptoms, really. But if you think about it, any problems that may cause the tree starving water, the leaves will start drying out from the margins with or without the yellow halo, and that can be confused as well with Xylella. But I would say that right now, if you go out and you look to the trees, you will think you are seeing Xylella everywhere, where it's not Xylella, it is the autumn colors, if that makes sense.
It makes absolute sense.
Because we don't have, be aware that at the moment we don't have Xylella here, and but as I said, the way the color breaks in the autumn is very, very similar.
Yeah. Okay. On a related question, David Skidmore's come up with a question. Given the long incubation period and similarity of symptoms of Xylella to other causes, at what point should a grower report these symptoms?
Symptoms should be reported as soon as possible. If you see any symptoms in any of your plants that you are concerned, don't ignore them. That will be my advice. Never ignore symptoms that you know in any plant. it may not be Xylella, it could be something else, or it could be something abiotic. Maybe it's nothing related to pests or diseases. But don't ignore any symptoms on plants. I think that will be my advice. Always better safe than sorry.
Yeah.
And also, we know that Xylella can be present on plants and never ever show any symptoms. And in some cases, will take very long time on showing any symptoms. But definitely, if you see symptoms, please report it.
So we'd rather have reports through Tree Alert than not to have reports through Tree Alert. So let us know.
Yes, I think for me, I always say there is never a silly report. You know, sometimes people don't want to report something because they think, oh, it may be nothing. Listen, in this case, I think I'd rather say to you, it's nothing, and that's it, that miss something that could be of relevance or of importance for all of us.
Perfect. Thank you. Okay, moving away from Xylella for a minute, one to ash dieback, and a question from Andrew Cook, who says that there doesn't seem to be much ash dieback in Wales from your map. There seems to be a lot more. Is it because it hasn't been reported? So.
Okay, that's right. The map that I show you, it was the map of the cases we have confirmed at Forest Research. Samples were sent to us to confirm these cases. Obviously, it is only people reporting through Tree Alert.
Yeah, well, from having done a bit of work with the Welsh government, I can categorically state that most 1 kilometre squares in Wales have at least one incident of ash dieback. So it's very widespread across Wales, unfortunately.
Yeah, the map was only reflecting the cases we tested in the laboratory. That they were sent to us.
Thank you, Anna. Now, a question on climatic conditions. So Lorraine has asked, is the series of mild winters one of the reasons that pests and diseases are seeming to increase?
Okay, some of them. It's like no, all conditions are good for all in a way, but yes, it's true. Having this in the last few years, we had very mild winters, and what we have seen is some of the pathogens that maybe they are not causing lots of damage in normal winter conditions, because we have several years of mild winters, we have seen an increase, and it's quite interesting because we see that the same, not the same species, but the same genus, you can see like a increasing of its presence on different hosts, and it's because it's mild conditions, and some fungi will do much better than others. Then yes, in some cases will help some pathogens, definitely. Yeah.
Okay. Yes, and certainly things like the spruce aphid is knocked back by very cold winters, and that's something that we're not seeing much of at the moment. Yeah. Okay. One from Andrew Cook, and you may not be able to answer this one as you're not an entomologist, and I'm not sure I can either, so I might call on David. But what are the predators of the frog hopper, and what other pests are vectors of Xylella? So the frog hopper is the vector for Xylella. So it's a...
Okay. Yeah. I do not know the answer, but I would like to say something. Obviously, people or some people get confused because obviously we don't have Xylella in the country, but the frog hopper or the leaf hopper is common in the country.
The cuckoo spit insect. Yeah.
It's not a pest. Okay. I just want to make this very clear. It's not a pest. However, if Xylella comes to the country, it will be transmitted by them. But at the moment, as I said, it's not a pest. It's not going to cause any damage. I don't know the answer about the predators of the.
No, I don't know. I don't know the answer either, I'm afraid. But the frog hopper for the audience is basically the cuckoo spit insect. So in spring and summer, you see these little bits of froth on plants, and if you move the froth, you can find these small green insects beneath the froth.
Okay. We call them a spittlebug.
Spittlebug, yeah, same thing. Yeah,
Spittlebug.
cuckoo spit insects.
We have, in some of the resources I mentioned, you have videos and you can see them, and you can remove the froth and you see them. But yes, as I said, I don't know the answer of the predators of them. Sorry.
Okay, you talked a bit about Bridget as well, the Bridget project. I don't know if you want to expand on that and other projects that you and your team are involved in.
I mean, with Bridget, as I said, we do not have Xylella in the country, but we know is in some countries in Europe. We know the problems is causing there, and what we thought is, can we get ready, or can we prepare? Can we gather some knowledge, intelligence that we can, if it comes, we are able to be better prepared? And one of the things we have been doing within Bridget is doing like a spittlebug survey, trying to find, where is the spittlebug and on which plants is the spittlebug feeding, for example, because if we have Xylella in the country, we know it's probably this insect that is going to spread the disease. Then if we know where the insect is and on what are the choices or the preferences for plants, then we may be able to know where the disease is going to go. Then within Bridget, we have different working packages, and one is, as I said, is looking into the spittlebug, and another one is looking to diagnostics and trying to improve the diagnostics on Xylella. Then we have also some monitoring of, in case Xylella comes, and people who have been working monitoring the disease in other European countries. We are preparing, apart from the resources that I mentioned, we'll have also some databases that they are open to anybody can search on these databases. They can look for different insects, and I think just have a look to the resources. I think they are very good. And as I said, is a consortium of different organizations, and we try to get ready or get prepared if we can. And that's Bridget. But I also work with Observatory. Observatory is a project that has been running now for quite a few years. And within Observatory, we work on a list of priority pests and diseases. Then we selected 22 priority pests and diseases. Some are in the country already, some have recently arrived, and some are not here, and we don't want them here. Then basically we have a team of over 200 volunteers who all around in England, Scotland, and Wales, and they just look to see if they can find any of these pests and diseases in the country, and they report them through Tree Alert. Then it's been very good in the last few years. We have very good reports, and we have been able to, for example, when Oriental Chestnut Gold Wasp, it was introduced to the country in 2015, and the second finding in a different area was by an observatory volunteer. Yeah. And that was very good. I mean, because what we did is, as soon as we detected this pest in the country, we did organize like a training session for all the volunteers, and within days we had the second report. Then that helped us to the monitoring of these pests and diseases, and it's like an early warning system.
Thank you. Yes, it's a really, really useful system, and should encourage people to get involved. It's a very good website to have a look at. The observatory website. Resources. Incredibly good resources that you can download and look at.
Exactly.
Yeah. Okay. Just a comment back on the froghopper or the spittlebug. Apparently, it can be a real pain on Potentilla fruticosa varieties or varieties of Potentilla fruticosa. So that's from Andrew Cook. And another quite relevant question, again to do with Xylella, but it also applies to other pests and diseases. If you spot symptoms of Xylella, or indeed other pests and diseases, that is not on your land, should it still be reported?
Oh, that's a tricky one. Okay. Tree Alert will allow you to report something that is not in your land. Okay. Obviously, if we have a report of, I'm not talking about Xylella now, I'm talking in general.
Yeah.
For example, any other pest or diseases that they are not in the country and they are quarantined. If somebody makes a report on a property that is not their own property, they can submit the report. I don't have the power to go and visit the site. However, if we look to the report and we are convinced that is a high probability that this could be a quarantine organism, the report is sent immediately to the tree health officers in the countries, either in England, Scotland, or Wales, and they will take it over. They will take action over this. But one of the first questions when you submit a report through Tree Alert, one of the first pages will ask you, are you the owner of the tree?
Yeah.
Are you submitting the report on behalf of the owner of the tree? Or do you know the owner of the property? We'll ask you these kind of questions. Then, yes, of course, if you see something, please report it. I have to say this, and I have to say, obviously, when you have a system to report pests and diseases, some people will try to report their neighbors.
Yeah.
Okay, then this is, it won't be the first time and it won't be the last one, obviously, but every report is checked carefully, and if the symptoms reported are of concern, the report will be followed.
Yeah. Thank you, Anna.
I don't know if that explains.
No, that's a good
Okay.
explanation. Thank you. Right, we've got probably about 7 minutes left, so time for a few more questions if people have any more to put in. There's one more that hasn't been answered yet, from David Skidmore. How has sequencing and PCR changed how you do diagnostics? And are more pathogens identified more commonly than before? Right, we've got 7 minutes, Anna. Go.
Go. Okay. First thing, we are able now to diagnose some of the common problems within 24 hours, and that is amazing. That means a sample coming in, we have a specific primers, we have real-time PCR with a specific primers, we can do a test and tell you yes, no, positive or negative, and that is great. Sequencing obviously has opened a new world, because what happens is some of the species, they were very close related, and sequencing is giving us the opportunity to distinguish the different species much better, and that is also fantastic. If we go to the high throughput sequencing, we don't use it in routine diagnostics. That is another conversation altogether. But what I can say is we do, in many cases, we have a specific protocols for diagnostics, molecular diagnostics on these pathogens, and these allow us to give very quick answers. And when you are dealing with an outbreak, that allows people during the operations to continue the surveys, because when you give a positive, they may have to initiate another 1 kilometer, 5 kilometer survey around this infected plant. Then having this quick answer, it really helps operations. And sorry, I was trying to go fast, but I hope I managed to. Okay.
Yeah, so that's quite an interesting point you bring up again about zonation around a potential outbreak. So I don't know if you want to talk a bit more about how you might deal with advise on an outbreak like that.
Yeah, I mean, obviously, okay. When you have an outbreak, obviously, every time you detect a positive, you initiate like a very intensive kind of survey, especially 1 kilometer around that positive.
Yeah.
And also you do a 5 kilometer as well. And then if you don't find any more positives, then that's okay.
Yeah.
But if you find another positive, you have to start again.
And
these the circles go, they can grow larger and larger.
Yeah, intersecting circles.
Yeah,
I used the word outbreak. I should perhaps have used interception, because we can intercept things, and if they're not established, it's not an outbreak.
Exactly. Then you have interception is before it has spread. Then if you detect something that is brought into the country but has not spread, it's an interception. But if it has spread, then you can call it an outbreak. And when you have one of these outbreaks, obviously the first thing that you do is starting having, like infected zone, buffer zones, and just try to limit the areas that they are infected.
So it comes back to tree alert, and why tree alert is so important is that we want to have things as interceptions which can be quickly nipped in the bud and don't establish rather than developing into full-blown outbreaks.
I mean, we did have one very in 2016. One of somebody reported chestnut blight on one side, and that side it was eradicated on that area. And since then we haven't had any reports in that area. I mean, we had reports in different areas, but not in that area. And it was a tree alert report, and immediately action was taken and eradicated. That's good news.
It is. We've probably got time for two more very quick questions. One more from Kate Burtonwood. You mentioned that Fraxinus americana and others were starting to show susceptibility to cholera.
Yeah.
What is a good resource to check the susceptibility of Fraxinus and other species?
Okay. What is a good
resource? Is there somewhere that people can find a list of affected species?
Not at the moment. There are different projects ongoing, and I have to say what I have shown here is the results of annual surveys that I do in tree collections, and we have a tree collection surrounded by Fraxinus excelsa that is infected, but it's an ash collection, different Fraxinus species, and what I have been doing is trying to monitor this collection over for the last 3 years, and these are the ones I'm trying to see if the disease is able to progress from the leaves to the stem. Unfortunately, I haven't published this, or it's not even probably. It is on our website, I think, but it's something that I think we should. I would like to publish on our website and update the list every year.
That's great. If you need help with that, let me know. And one final question, again about tree alert. Can shrub pests and pathogens be reported on tree alert? And if not, what is the best way to get diagnoses?
Okay. If it's not a tree, obviously the first thing I would recommend always is if you are a member of the RHS, obviously go to the RHS because you have a service free to members there. If you can always contact obviously FERA at York for diagnostics. In the case of Xylella, we made an exception, and we did say if we suspect Xylella, please use tree alert. And we work because your report will be sent immediately to the animal and plant health inspectors, APHA, and they will follow it up. Then tree alert is not standing there on its own. We are connected, or we work together with the different agencies. Then for the case of Xylella, as I said, if you have a shrub or a lavender, rosemary, or whatever you think could be Xylella, you can still report it. Your report will be sent to APHA, to the Animal and Plant Health Inspectors, and they will be in touch with you.
Thank you. Well, I think we're at 6 o'clock, so that's time. And can I thank you, Anna, for a really interesting presentation and for fielding the questions so well this afternoon. If people would like more information, there is information on the projects that Anna's talked about on the Forest Research website. That's www.forestresearch.gov.uk. Otherwise, feel free to email us with questions that you might have.
Okay, thank you very much.
Thank you very much.
Okay, thank you.